the same city, the same 1,000 restaurants, the same KNN graph connecting each restaurant to its geographic neighbors. But now, instead of ignoring the edges, we use them. The edges carry information. Focus on the Chinatown Italian. Its own menu says Italian, but along each edge, its neighbors' menus flow in. Edge 1 brings dim sum features. Edge 2 brings Peking duck features. Edge 3 brings wonton features. Banksy creates a combined profile, the restaurant's own menu plus a weighted average of its neighbors' menus. The combined profile says, Italian food in a Chinese neighborhood. The formula is simple, self features plus lambda times the mean of neighbor features. Lambda controls how much the neighborhood matters. Set lambda to 0, the edges disappear. Only the restaurant's own menu counts. This is pure Seurat. The two Italian restaurants have identical profiles. Set lambda to 0.3, edges appear. 30% of the profile comes from neighbors. The profiles start to diverge. Set lambda to 0.7, edges glow thick. The Chinatown Italian's profile is now dominated by Chinese neighborhood features. The biologist tunes lambda based on how much spatial context matters for their tissue. With Banksy's neighborhood-aware features, the clustering changes, side by side. On the left, Seurat clustering. Both Italian restaurants are red, same cluster. The KNN edges are drawn in grey but weren't used. On the right, Banksy clustering. The Chinatown Italian is now orange, a new cluster defined by Italian cuisine in a Chinese neighborhood. The Little Italy Italian stays red. The edges cause the split. In biology, tumor-adjacent macrophages cluster separately from stromal macrophages. Banksy doesn't just refine cell type labels, it discovers spatial domains. Zoom out to the full city map. Chinatown Food District, a region where Chinese restaurants dominate, and even non-Chinese restaurants are influenced by the neighborhood. Little Italy District, Financial District Lunch Zone, Waterfront Seafood Row. These domains contain mixed restaurant types, but share a neighborhood character. In tissue, the tumor-immune interface, the perivascular niche, the fibrotic border zone, the recommended pipeline. Seurat first, normalize highly variable genes PICA. This produces clean expression data. Banksy second, compute spatial neighbor features combine with expression using lambda, cluster on Banksy features. Ampere, Seurat clusters answer what cell types exist. Banksy clusters answer what spatial domains organize the tissue. Both are valid and complementary. Report both. Two Italian restaurants, same menu, different neighborhoods. Seurat tells you what a restaurant is. Banksy tells you where it belongs. The menu defines the cuisine. The neighborhood defines the role. In spatial transcriptomics, both matter. A macrophage is always a macrophage, Seurat's answer. But a tumor-adjacent macrophage plays a different role, Banksy's answer. The graph was always there. Banksy finally reads the edges.